Hello, my name is Roger Williams. I'm the Assistant Director of the Microbiology and Immunology Undergraduate Department here at the University of Miami. Today I'd like to go through with you the teaching assistant setup we have in the front of our Introductory Microbiology and Immunology laboratory. The laboratory runs concurrently with the Introductory Microbiology and Immunology course, which is offered in the spring semester of every year. First, I'd like to go through with you the scope, the particular scope we use in the lab. We currently use the Nikon E200 clinical microscope with a few changes. We've replaced the head of the scope with a trinocular head. We've added a wide TV tube, in addition to C-mount and a C-mount compatible USB microscope camera. The Trinoc has three settings. It has a binocular setting, which allows for the image to pass through the eyepieces, a binocular and photo setting, which allows for part of the image to be sent to the eyepiece and part of it to be sent to the camera, and then lastly a photo setting that sends all of the image to the camera. For today's demonstration, we will be setting it at the mid portion, or the mid setting, where part of the image is sent to the eyepiece and the other part to the camera. So first what we'll do is we'll connect the power to the microscope, and we'll also connect the USB for the microscope. And then we'll wait for the camera to be recognized by the PC. And which it has. The light is now lit, so we can now initialize the software for the camera. I'll select the camera that we're using. The preview screen for the software will appear on the PC screen. Since I'm using a dual monitor setup, I will now take the preview screen and send it over to the secondary monitor, which now allows it to show up on the lab monitor. So right now, the field of view is pretty dark. So I'll now turn on the microscope, and as you will notice, the screen lit up, and right now we have in focus a fairly large parasitic organism. As you can see, as I move the stage adjustment knobs, we can run down the entire length of the organism. And this is pretty much, this is in real time, so you can notice that there's relatively no lag at all in the controls. And we can do anything with it, adjust the lighting, whatnot. So another feature of the software, the camera software, is the ability to capture an image. So right now I'll show you that. And it's pretty simple to do. We basically all we have to do is select the capture, and what's going to happen is that we'll be able to then take a snapshot of what it is that we're looking at, and then this snapshot we can upload to a number of student sites. One for instance being Blackboard, so that students can use the images to study for practical exams, quizzes, and other types of graded activities. So a very valuable tool for the student, and also the TA can utilize this software in order to shed light on a particular organism. He or she wants to, for the students to try and find, maybe in the lab. He or she could put the image up on the screen and it would give the students an idea of what they were looking for, so that it would aid in their search. You know, and just a number of uses for this sort of application. So very useful for the student as well as the TA. In addition to that, the instructor, as you know, there are various resources out there on the web and whatnot that a TA could make use of in the lab to instruct his or her students. So it's very easy to pull up a website and display it for everyone to see in the classroom. In addition to that, we've eliminated chalk and talk type teaching experiences and have gone to strictly PowerPoint presentations in our laboratory. That also is very very streamlined, and all the students can view the presentation, which frees up time for the student, because he or she doesn't have to be focused on trying to copy down every bit of information that's being lectured on. And also frees up time for the instructor, because the instructor can now take less time writing down the exercise for the day and whatnot, and frees up a lot more time for instruction, actual instruction in the laboratory. So these are just a few of the many features and instructional aids that the TA in our microbiology and immunology lab has available to them for their instruction.